Okay, here's part 4, and now we're talking about putting the report together, and that's probably where you'll have a lot of questions. Okay, so let's go over the basics here. So this is all stuff you need, and this is me telling you basically what it can do and how you can use it. You may not need every single thing here, but you'll use a lot of it. So you're looking for good information for every gene, and there are different ways to get that, and if you get good information from the first test you do, you don't have to do the rest of the tests. So let's go through these sections. All right, so we already did. The last section was all about mapping. And so your job is to take those blast results and map it out in a way that has all the information on it, including the e-values and bacteria and what genes you found and what direction they're going, and make a little information graphic with a figure legend underneath that. And so that's going to be used somewhere inside the paper. And the one I made is much better than the one in the example. That's actually the weakest part of the example is their map. All right, section 2, bioinformatics. So this one we've also almost completed. So bioinformatics means finding information, and so we're going to go through we're going to have a hierarchy, if you like, of information. And so let's make a new little document here. And so step 1, the best possible information, or the easiest, is going to be if we can find information from protein blast. And boom, if we get a hit with an e-value of less than or equal to 0.01, then report, and here we go, and it is to a gene of known function, report it, and you are done. So there you go. Yeah, so you look at these results, and yeah, bam. If for every gene, for all, in my case, 13 genes, you're going to do a little report, a little write-up. And so let's go through here. So for gene 1, here's what we found. We found nothing. Oh, okay. And so we failed at criterion number 1. Protein blast gave us nothing. And so the second thing we can try is HH-pred. HH-pred. And if we get a hit with an e-value of 0.01 or lower, and it's a gene of known function, then we're going to report it again, and we're going to be done. So we're just going to copy all of that. And HH-pred also has e-values, so marvelous. So we can go back here since I didn't copy my proteins. There we go. And it's got all my proteins in order. And so there it is. That's the first one. So let's just copy that. Copy. Let's go over to HH-pred. Now this is a really good program. It's very good at finding difficult, distant matches. It occasionally gives you false positives. It'll say something is a match when it really isn't. And, oh, I see why we've got some problems here. So just looking at this thing, just sort of sequence gaze with me, if you will. I'm looking at that and I see a lot of the same letters over and over again. I see a lot of G's, a lot of glycines, GGG, GA, GAG, GGG, GG. And glycines indicate flexibility. So this is a protein that would be very bendy, very floppy if it is really a protein. The other thing I see here are lots of arginines. Oh my goodness. R's up there, and histidines. And so what we've got is a super basic charged, very flexible protein. And things like this exist in the world, but it is very difficult to assign them a particular function because they are going to be used in different ways at different times, and their function is to be bendy and positive, and usually to stick to something. And we won't know exactly what this thing sticks to without a whole lot of extra actual lab work. But let's give it a go. So we're going to run this thing through HH-pred. We'll submit the job, and it will just run here for a sec. And HH-pred is going to take a little longer than BLAST. It'll sometimes take 2-3 minutes, but yeah, but it'll work. Okay, so if we get an HH-pred hit, and I don't think we're gonna, but if we do, that's fine. We're gonna report it as long as the e-value is good, less than or equal to a 1%, 0.01 chance of being wrong. And so what if both of these things fail? So if both of these things fail, then your last resort is just both of the tests above do not give an answer. You just describe the protein and you're done. And so the protein, we're going to describe it with in terms of molecular weight. Okay, so boom, there is our hit. The color scheme on HHPred worked just like the one on Blast. And so a hit that looks cool color or black is probably not going to be reliable. And how unreliable, you say? Well, it's got an E-value of 460. So 460 out of 461 times that you get a hit this awesome, it's going to be wrong. And that is also the only hit that it gave us. And yeah, I'm looking at that and thinking, no, I don't believe it. I do not believe that at all. So for this particular gene, HHPred has not been successful, but that's okay. We've still got a further fallback. And so it's one of our other tools here. Let's go. Boom. Okay, so we've already done BLAST, Open Breeding Frame Finder, and HHPred. And so our next one is going to be the Protein Molecular Weight Calculator. And so let's just go there. Protein Molecular Weight Calculator. And boom. And there's a bunch of them, and they all work just fine. Okay, and we're just going to paste our single protein in there, and you got to check that you only put the one in there, and then submit. And it says that it is 205 amino acids, and it is 21.93 kilodaltons in size. So that's fine. 205 amino acids, and so we'll just say the number of amino acids, and predicted molecular weight of one molecule, which is going to be 21.93 kilodaltons. This is going to be in kilodaltons. Boom. And yeah, that is our ultimate, if we can't, like if your mother told you, if you can't say anything nice, don't say anything at all. Well, in this case, if you can't say anything interesting, you're going to tell us the number of amino acids and the molecular weight of your potential protein, and that's going to be good enough. So there we go. That's our new maxim. And I will post this up online. That's what you're going to do for that section. So bioinformatics is going to be going through all these things. It's the same sort of information that is on the map, but you're actually going to go through one at a time. And then you are going to use your noggin and tell me, so what are the methods that you used to get these? And so you can group all the ones you found with BLAST together and all the ones with HHPred together, whatever. Describe your methods well enough that another scientist will be able to replicate your work. And so that is like saying that we used Protein BLAST, but we've changed to the NR, non-redundant database, because that's a thing that we did. And then you're supposed to discuss whether or not the identification should be considered reliable based on the stats. And so the answer will differ for each gene, but you're going to cover all of the genes in a very succinct, very small sort of way. And so here's our proteins over here, and if we go back to our recent results, there it is. Okay, so the first one, yeah, so for the first one, I would say we should not consider this identification reliable because, yeah, because it's junk, basically. It gives us no hit on Blast. If we went to one of these, that gives us a very nice hit. Then we could say, yes, that's reliable because it has an E-value of 0, which is indicative of a very high confidence. All these things are, see, that's the thing with science. You can never be right or wrong, and there's no such thing as an exact fact. All there are things which are likely to a different degree. And so we would never say anything is true, but we would say that the chance that we're wrong is less than 10 to the 1 in 10 to the 180th power. And we would say that's good enough that we can say we're probably right. But we would always speak in terms of statistics, in terms of, how certain are we of a particular conclusion. And that's what you're going to learn to do with this paper. So you're going to say that, yeah, we have a high confidence in this because the E-value is 0, which is less than 10 to the negative 180th power. And so it probably is a PAS domain containing sensor histidine kinase. And yeah, and you don't have to talk about what organism it's from. That's going to be what the next person is going to do. So boom, that's the mapping done. That's the bioinformatics done. On to the ecology. Ecology and adaptations, these two sections are both kind of like book reports. And so you don't even have to do the thing that we've done here if you want to just use the results from the bioinformatics person. So if you guys are on speaking terms, just talk to your bioinformatics person or your mapping person and ask them, where is all the data pointing, all the good data? Now, we're not talking about that sort of iffy data that's in the middle. We want the really good, error value of 0 type data. And if it's all pointing toward Ralstonia solanacearum, then bam, that's the thing that you are going to use. The other thing that you could do, if you want to confirm this yourself, is you can use a thing called nucleotide blast. And here, let's show you that. So let's go to another blast page. Boom. And let's get our little document up here. We need bacterial genome. So there's my section. It's still copied, or it's still highlighted, so I'll just copy it again. And all right, let's go nucleotide blast this thing. So instead of doing protein blast, which is over here, we're going to do nucleotide blast over here. And this just compares DNA to DNA, like for like. And it automatically defaults to the NR database, and that's fine. Let's just hit blast. And this will take, I don't know, a couple seconds to come in, and then this will tell us what has DNA similar. Now, DNA match is only going to work for things that are very, very, very similar. Even if you have different species in the same genus, you sometimes will not get a good DNA match there. And so, we're going to use our same criteria, and we're going to look through here and say, all right, what is this, and why do we believe that? So I see a bunch of answers here that all have E values of 0.0. And yeah, actually, pretty much everything on here has an E value of 0.0. Let's look up the ones at the top, though. And so these have 99% identity. And so that would be a stat worth mentioning here. And okay, so 99% identity to Ralstonia solanacearum, or 99% identity to Ralstonia solanacearum, or blah, 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 blah. And so it looks like the top couple of hits are all Ralstonia solanacearum, all with E values of 0, all with 98 or 99% identity. And so with that information under my hat, I'm willing to say that, yeah, this thing that you've got is most similar to Ralstonia solanacearum. You can never say it is anything because we don't know. It just has this little part that you're looking at looks like it either is Ralstonia or it was stolen from Ralstonia, and bacteria do that all the time as well. But we're going to say here's why we're confident. We're confident because the E values are 0, and so yeah, Ralstonia solanacearum. And so from that point, once you've got that one little statistic and that one type of bacteria, Ralstonia solanacearum, most likely, so bam, now you're ready to go. And so you're going to choose Ralstonia. Let's just work with Ralstonia since all the top hits were to that. And let's research its ecology, where it lives, what problems it causes, and how it gets its energy. And you can use things like Wikipedia just to get started on this. That's okay. So let's see. Ralstonia solanacearum. There we go. And it is a soil-borne bacteria. And the one that we're using was a problem in a banana plantation in the country of Colombia. And there we go. And so there we go. This is the thing it is. It's in the genus Ralstonia, which is part of this other group of things, and up to domain and phylum, etc. And this is what it does to tomato plants and banana plants and everything. It just causes them to wilt and die, and the veins of the plant will sometimes go cool colors, indicating death. Oh, one of the world's top 100 worst invasive species that move around from country to country. So, yeah, that's pretty bad. So great, that's telling us a little bit about it, and that's fine. Now, what a pro would do is go down here to the references. And so, there we go. It's giving us some nice references, and you would go and see if you can read those references. So like here, it's a complete genome sequence of Ralstonia, one that kills potatoes. And bam, now you're in a paper, and now you're in actual data that you can cite. So don't be citing that crummy Wikipedia data. Cite some of this good stuff. And so this is a journal that just deals with genomic sciences, so genome sequences. And let's have a look at it. So when you read a scientific paper, you're going to start with the abstract usually. And so Ralstonia causes bacterial wilt in potatoes. Yeah, okay, that makes sense. And Ralstonia solanacearum, that's the species. Strain UY031, that's just the particular isolate, belongs to this particular group that people have categorized before. And yeah, they use terms like race and biovar. These are the real throwback people. Still use these for plant science. But yeah, most everybody else has moved on in the genomic era. And so this paper, they are dragging those old fogies into the genomic era. So the completely sequenced genome for this thing is the first complete genome for this particular type, which is great. And so what did you find? Well, we found a bunch of type 3 effector genes. And so you can say, okay, what is that? What is a type 3 effector genes or effectors? And then bam, sophisticated bacterial virulence factors. Fantastic. It is a type of secretion system, and yeah, oh, that's interesting. So then you can look up what a type 3 secretion system looks like. And there, bam. It is a giant machine that comes with an ATPase and a bunch of pumps. And these bacteria have multiple membranes around them. And so they will actually, so these are the two bacterial membranes, the inner and outer membrane of the bacteria. And they will actually be like a little channel, almost like a needle that you can send other proteins, other toxic proteins. You can just punch a hole into a cell. So this is the plant cell's plasma membrane. And you can just squeeze out these proteins, and they're like little, it's like injecting the plant with poison. So it'll die and release all its delicious nutrients, stuff like that. So those are a cool thing that you could talk about in Ralstonia. Or you just go into Google Scholar, scholar.google.whatever, and let's say Ralstonia solanacearum, boom. And there we go. We got a whole bunch of them. So there's the first sequence of Ralstonia. It occurred in the journal Nature, which is really prestigious journal, but a long time ago, 2002. That's back when you could get a Nature paper for one genome sequence. And so it says it's got two parts. Interesting. So it's going to tell you about those two parts in here. And yeah, it's going to tell you what sort of genes they find. So on this Ralstonia, which I think turns out to be common for most of them, they actually have a chromosome that's 3.7 million bases, 3.7 megabases. And they also have like a second chromosome. It's almost like they have two chromosomes. And they call the second one, because it's smaller, they call it a megaplasmid. But really, you should just say, boy, that's another chromosome. But I think the main chromosome has everything it needs to live, and the megaplasmid, it can survive without that, but those are all the genes that help it be extra pathogenic and kind of toxic and hurtful to plants. And so if you just go through this paper, and whenever you come up with a word that you don't understand, so pathogenicity, and let's say we don't know what that is, you're just going to look it up. And bam, ability of an organism to cause disease. And they say, oh, okay, fine. And yeah, write this up. And every time that you cite a fact, I want you to cite the reference that it came from. So one of these references would be this thing. Boom. Yeah, that particular paper in Nature, volume 415, page 497 to 502, from 2002 by these guys, Salno Ubat. It's et al., and that's the title of it. And there we go. And so this is a good way to do references. So they've done this right here. So they're talking about Ralstonia solanacearum, and then they want to cite two papers about Ralstonia solanacearum. And so they just put these little numbers there, and you can do the same thing. Just do it manually. And each number refers to a reference. And so there is the reference. It's popping up because this is a nice little hot link, and Nature's a prestigious journal with good coding. And there's the second one, and so yeah, boom. It's talking about what is Ralstonia, and yeah, what does it do? And looking at the genome is a really good way to figure out what it does. But there's going to be lots of other papers on here, and you want to make sure that you're finding them through Google Scholar. That is a very nice way. And so again, that web site is scholar.google.com. And boom, that'll take you. Oh, you gotta spell it right though. There we go. Boom. And then Ralstonia solanacearum, etc. If you want to know about Ralstonia solanacearum induced wilt, bam, there's going to be papers on that. If you want to know about pathogenicity, there's papers that are telling you how it goes about picking this up. So generally for a section like this, you want to draw from somewhere between 5 and 10 of these papers, 5 and 10 different sources. And that's going to be a good section. Yeah, you're going to get a lot of good information there. And you're talking about, I don't know, maybe 2/3 of a page or half a page for yours, but that's like single spaced and really packing it in. Okay, and that's going to be the ecology section. It is telling me about Ralstonia and what it does and how it does it. Now the adaptations section is, this is my favorite section. And so, up here, you're going to get in contact with your bioinformatics person. If your bioinformatics person is sleeping on the job, 1, kick their ass and wake them up. And 2, that's fine. You're going to have to do their work yourself to figure out what you should do. But you can use Open Reading Frame Finder to find all your proteins. And then you can use either BLAST or HHPRED. It just says HHPRED on there, but you can use BLAST as well. That's fine. Whatever gives you your thing. So Protein BLAST is the #1. If it works, great. If not, HHPRED is there for you. And if both tests fail, boom, molecular weight. So adaptations, you're the first person that gets to kind of pick and choose. So ecology, they can pick and choose which part of the bacteria or its disease or whatever that you want to talk about. On adaptations, you get to pick some of the genes that you found. And so, for example, on my section, if we go back here, so I've got that sensor histidine kinase with a PAS domain. And right next to it, I think, I have the other part of the 2-component system. Yeah, and so that would be a good thing. And so once again, you're going to go to Google Scholar. And so this is a PAS 2 histidine kinase 2-part sensor. And bam, there we go. We find out about him. And so you're going to read papers about histidine kinases, but you pick one. Google a bunch of the things that you found. See if you can put a couple of them together and make a story. This is one for somebody with a nice big brain and willing to go out there and do some legwork. It's not quite as cookie cutter as the other sections. But yeah, you're going to pick whatever is a good story and is going to fill about half a page to 2/3 of a page with really interesting detail. That's going to be a great choice. So here we go. And if we didn't want to do that one, we could go up here and pick one of these, for example. And so methionine formal transferase, that's great. So that's involved in translation and making the tRNA. And then here we have a thing that helps modify the 16S ribosomal RNA. So we've got 2 genes in a row that are involved in the same process, which is generic translation. But this is a good opportunity to talk about how these 2 proteins fit into generic translation. So if you didn't want to go with the 2-component system here, you could go with these 2. You should have more than 1 gene. You shouldn't just be talking about 1 gene. It's got to have at least 2 in there. But yeah, find 2 that you can make some kind of story about. And somewhere in all your genes, there are going to be 2. And they'll probably be right next to each other that have something to do with each other. And yeah, just do your best. And in the end, if you can't figure out which 2 go together, all right, just find 2 that are next to each other and tell me about both of them. And yeah, there will be a connection that you're probably missing, but that's okay. At least you're doing the research, at least you're telling me about it, and you'll still get a good grade. Because this is just about researching these things. So let's learn a little more about this very specific gene, oh my goodness, through Google Scholar. So boom, boom. And we don't want the one from Ralstonia. We want to know what this gene does in general. And so tRNA, ooh, interesting, okay, fine. So there's a tRNA methyltransferase in yeast. So what does this thing do? And so this will probably tell us about its sequence, might even tell us about its structure. And so you can read that and see what they did and how they did it. The other thing I would say is when you're reading these papers, and let's see, so it may be a good idea to leave off some of these things. So let's get a little less specific. Methyltransferase of the type RSMB. And there we go. We got an RSMB coming up down there. RSMB signature motifs. There we go. Several RSM methyltransferases like RSMB. So each of these things uh, is talking a little bit about our gene. Maybe talking about other stuff as well, but let's see. So let's find RSMB. There we are. Okay, I've been identified as a B, and there, this paper has now helped us out. So it says there are two papers about RSMBs, paper number 2 and number 8. So let's just drive down. The references are always at the end. There we go. Number 2, bam. So we can look for that thing. And number 8, bam. Okay, they're both in 1999. They're both in biochemistry. So, all right, let's just pick the first one for now. There we go. Copy that. And we'll go right back to Google Scholar. Let's put that in there, and then bam. Okay, so that's taking us to the actual paper that's about this RSMB, probably the first time it was ever discovered. And there it is. That is talking about it. And so it's saying that this is a thing that has homologs. So there's the same gene doing the same job in prokaryotes, that's like bacteria, archaea, they sort of look like bacteria, but they're different, and in eukaryotes. So what we found here is a very fundamental gene. It looks like everything that's alive has ribosomal RNA, which is true, and it looks like that ribosomal RNA needs to be methylated in order for that thing to stay alive. And so let's see if we can get the full text of this one. Some you will, some you won't, and it looks like this one is paywalled. Yeah, this one's paywalled. So, but we can take a little bit of information here from the abstract and we can cite that. And that's going to be good enough for this one. So you can read through there, and there's still plenty of information packed into this nice little abstract. And so that's going to be another way to do section 4. You're going to find a story about 2 or more of your protein, and then you're just going to talk about a couple of your proteins. If all your proteins are involved in the same process, you can talk about all of them, but at a minimum, I want you to at least hit 2 of them. Tell me what they are, what they do, and how they contribute to the organism, basically keeping it alive. So those are the 4 sections. A map, the bioinformatics, using all the different methods one at a time to go through until you've exhausted all the options for matching this to something. The ecology, where you're probably going to be telling me about Ralstonia solanacearum, although if your sequence says something else, then you tell me about something else. That's fine. And then the adaptations section, where you're going to be talking about some specific genes in here, and you're going to be doing a deep dive. What's their function? What's their structure? What do they look like? What do they do? How do they help the organism stay alive? And those 4 sections together are going to be single spaced, and they should take up no more than 2 pages, and that's including the little information graphic. If you want to show a picture of a sick plant or something, put that in an appendix, stick it on the 3rd page. I want 2 pages of packed high density, text with a bunch of numbers in it and all kinds of scientific detail that a regular layperson just would not understand. That's the mark of a really good paper. And so that's going to be all for this section. Thank you very much. And yeah, that's it. Bye-bye.